Hey everyone, and welcome back to Mastering Medicine video lectures. Today we're gonna learn about cell membrane structure and its functions. During this lecture, first of all, I'll talk about cell membrane models. Okay, it means different structural and functional designs that were proposed by different scientists as time was passing. For example, some scientists came in this set that cell membrane has a lipid nature. Other scientists came in this set that cell membrane is made of two layers of lipids, or it's a bilayer model. Okay, and other scientists came in this set that cell membrane has fluid mosaic model. That I'll explain each of these one by one. And then after this concept, I'll talk about basic structure of a cell membrane and its related three important components. For example, a cell membrane is made of lipids, it's made of carbohydrates, it's made of proteins. I'll talk about each of them one by one. And then at the end of this lecture, I'll talk about important functions of a cell membrane. Okay, so let's dive in. First of all, let me talk about different cell membrane models. I'll start from basic cell membrane model or basic cell membrane design that were proposed for the first time, and I'll go through all different designs one by one up to fluid mosaic model. Okay, for example, the first person or the first scientist who came and proposed a type of cell membrane model that he said that cell membrane is made of lipid nature. Okay, and he did an experiment. His name was Master Verton. So let me explain this experiment. Okay, for example, he took some small cells that were related to the root of a tree. Okay, and these cells are called root hair cells. These are called root hair cells. He extracted each of these cells one by one. Okay, and then he put these different hair root cells in different mediums and solutions. For example, some solutions were lipid soluble, some solutions were water soluble, some solution was made of salt, some was made of sugar. Okay, so there were different types of solutions. And finally, after this experiment, he examined each cells one by one. He found these cells that were in lipid solution. So these lipid soluble things were passed so easily through the cell membrane into the cell. Okay, for example, he found that lipid soluble substances can easily pass the cell membrane. Okay, so based on this theory, he said because lipid soluble things can pass easily through the cell membrane, so because of this concept, it might be the cell membrane might be lipid nature. Okay, so he said that cell membrane is made of a lipid structure. Got it? And this was the first theory about the cell membrane, the first model that a cell membrane has a lipid nature. After him, another two gentlemen came. Okay, they said that Master Golter and Grendel, they proposed another theory. They said that yes, cell membrane has a lipid nature, but cell membrane is made of two layers of lipid. For example, this is a cell membrane. So this cell membrane is made of two layers of lipids. Okay, So they did some experiment. Okay, they chose an RBC. Okay, they chose an RBC. They put this RBC in a solution or in a medium to destroy this RBC. So they destroyed RBC and they divided this RBC in different parts. Okay, then they extracted lipids from the cell membrane so carefully, and they put and spread out all these lipids as a one layer, as a monolayer over the water. Okay, on the surface of water. And they chose RBC because RBC doesn't have organelles, it doesn't have nucleus and so on. Okay, so to measure its surface area, it was easy for them. Now, when they spread out all these lipids over the water or on the surface of water in a special divisor, and then they compress them, all of them together. Now they measured this surface area so carefully. Okay, when they measured this surface area of lipids on the surface of water, and they compared this surface area with the surface area of RBC, this length of the surface area was double as the surface area of RBC. So that's why they said, because it's double compared to the surface area of RBC, so the cell membrane might have two layers of lipids. And also they added a little bit details to this. They said that this lipid has two parts. Its one part is, or its head part is, a hydrophilic. It loves water. And its tail part is hydrophobic. Okay, the tail parts try toward to the air, and the head parts try toward to the water. Okay, and they said that, for example, this is a cell. Okay, this is a cell. So we have water inside of the cell, and also we have water outside of the cell. Now they made a design. They said that, for example, it's two layers. So this part of lipid, which is the head part, it's hydrophilic. Okay, and this tail parts or feet parts are hydrophobic. Okay, so the another layer of lipid, its head part is also hydrophilic, and because we have water inside of the cell, and its tail part is hydrophobic. Okay, so these two tail parts try to work to each other, or they try to be in the middle, and the hydrophilic part try to be to the water area. Okay, and they made this model that, for example, this is the outside of the cell, this is the inside of the cell, and between inside and outside we have two layers of lipid, and they're designed like this, that the tail parts are trying to be in the middle, and the head parts are trying to be to the outer and to the inner side of the cell. That we'll talk more about it later on, that it's a phospholipid. Okay, so for the first time they proposed a lipid bilayer design. Okay, and after these two gentlemen, another gentleman came. Okay, and his name, or the name was, okay, Master Danieli and Devson. Master Danieli and Devson proposed another design for the cell membrane. They said that yes, cell membrane has a lipid nature, and they said yes, cell membrane is made of two layers of lipid, plus a cell membrane has another cover from the inside and from the outside, another cover of protein. Okay, and it's like a sandwich. For example, this is a sandwich, and these sandwich particles are between the two layers of bread. Okay, so these two layers of lipids are between the two layers of proteins from the inside and from the outside, and they proposed a sandwich model. Okay, and later on now we know that yes, it's a sandwich model because we have a lot of integral and peripheral proteins from the inside and from the outside of the cell membrane, or to the inside and to the outside of the cell membrane. Got it? After these two gentlemen, another gentleman came, and his name was Robertson. Mr. Robertson said that yes, cell membrane has a lipid nature. Yes, cell membrane is made of two layers of lipids. Yes, the cell membrane is like a sandwich. Okay, it has a cover of protein from the inside and from the outside. Plus, he said that cell membrane is made of three layers. Okay, first of all, he studied different cell membranes of different cells related to different living beings. Okay, and he said that all cell membranes related to all types of living beings are made of three layers. Okay, or he proposed a model which is called unit membrane. It means that all cells of different species and living beings are made of three layers. When he studied it by microscope, he found that the inside, this inner, the middle part between the outer side and inner side of the cell membrane, this middle part was light under the microscope. It looked and appeared light under the microscope because it's made of fatty acid chains, and the outer and inner side looked and appeared a little bit dense, okay, under the microscope. So this is one layer from the outside, this is another layer from the inside, and this is another layer from the middle part of the cell membrane. Got it? So he proposed trilaminar model, or three-layer structure of a cell membrane. Okay, and after all of them, two gentlemen came and they proposed a new model which is called fluid mosaic model, that we will talk about this in great detail. They said that a cell membrane has a mosaic structure. Do you know what is mosaic structure? For example, when we make an art or a picture by beautiful stones, okay, by placing of different beautiful stones, so this art is called mosaic art or mosaic picture. So because cell membrane has a lot of beautiful proteins across them, from the outside of it, from the inside of it, so he said that it has beautiful stone-like structures which are proteins. Okay, and also they said that cell membrane has fluidity, that I'll explain it later. Fluidity means that cell membrane has a little bit laterally movement. These fluids or these lipids are movable laterally, and this lateral movement of lipids are called fluidity. Okay, so now we will talk about the basic structure of fluid mosaic model. That a fluid mosaic model is made of lipids, proteins, carbohydrates. We'll explain all parts one by one, and then at the end of this explanation, I'll explain some specific characteristics of fluid mosaic model and its related terms. Okay, and then we'll talk about function of a cell membrane. So let's dive into the fluid mosaic model. Now, let me talk about chemical composition of a cell membrane. A cell membrane, first of all, do you know what's a cell membrane? A cell membrane is a narrow border around the cell. Okay, we have two terms: plasma membrane and cell membrane. Um, Cell membrane is general. It uses for the membrane of a cell. It uses for the membrane of organelles and so on. And plasma membrane is a little bit specific, and it uses to the membrane of a cell which surrounds the plasma. Okay. Now, anyway, a cell membrane is basically made of three chemical composition. It's made of lipids. It's made of proteins. It's made of carbohydrates. Now let me talk about lipids of a cell membrane. First of all, important lipids. A cell membrane is made of phospholipid, that we will talk about it now. And then a cell membrane is made of cholesterol. A cell membrane is made of sphingolipids, and a cell membrane is made of glycolipids. So let me talk about phospholipids. That what are phospholipids? So easily. First of all, a phospholipid is an amphipathic lipid. It means phospholipid has two sides. Its one part is hydrophilic and its another part is hydrophobic. Its one part loves water and its another part repel the water. It doesn't love water. Okay, so now let me explain it. The basic structure of a phospholipid is that when it's a type of complex lipid, this is a glycerol molecule, or this is an alkyl group, and a glycerol molecule has three carbons. This carbon number 1, this is carbon number 2, this carbon number 3. When we add two fatty acids to first and second carbons of glycerol, okay, this is one fatty acid, this is another fatty acid, okay, plus when we add a phosphate group to the third carbon of glycerol as a collectively glycerol molecule plus fatty acid plus two fatty acids plus phosphate as a collectively, all of them are called phosphatidic acid, which is the basic backbone of phospholipid. And when we add an additional group to phosphatidic acid, for example, when we add nitrogenous bases, for example, when we add choline, okay, or some other nitrogenous bases that we'll talk about them later on. So as a collectively, phosphatidic acid plus nitrogenous base, all of them are called phospholipids. All of them are called phospholipids. We have different types of nitrogenous bases that are usable in phospholipids structure. Okay, for example, we have a nitrogenous base which is called choline, and we have a nitrogenous base which is called serine. We have a nitrogenous base which is called ethanol amine. Okay, and when we add these different nitrogenous bases to phospholipid, so the name of them will be changed. They're called phosphatidyl choline, they're called phosphatidyl serine, they're called phosphatidyl ethanol amine, and so on. Okay, now anyway, now this is a phospholipid. For example, this is a phospholipid. Its head part, this is the head part of phospholipid, and the head part of phospholipid is made of glycerol. It's made of glycerol, okay, plus it's made of phosphate, plus it's made of a nitrogenous base. So as a collectively, this head part is water soluble. It's hydrogenous hydro a hydrophilic. Got it? And the tail part or the feet part of phospholipid, which is made of fatty acids, they're hydrophobic. Okay, they repel the water. Okay, they're hydrophobic. So this is very important. Now, in a cell membrane that I've explained it previously, in a cell membrane, okay, we have two layers of phospholipids or two layers of lipids, okay, and they arrange like this, that the head parts are trying to be to the water area. Okay, for example, outside of the cell we have water, and inside of the cell we have water, so they try to vert to the water because they are hydrophilic, and the tail parts are trying to vert to the middle part of the cell membrane, and they are hydrophobic. Okay, so this is the net arrangement of phospholipids in a cell membrane. This is the outside of the cell membrane, this is the inside of the cell membrane. These reds are a head part of phospholipid, and these are tail or feet part of phospholipids. Got it? Now, let me add a little bit detail to it, that in most cell membranes, we have two leaflets. Okay, in a cell membrane or in a fluid mosaic model, we have two leaflets of the cell membrane, the inner leaflet and the outer leaflet. The inner leaflet is made of phosphatidyl serine, and phosphatidyl inositol, and the outer leaflet is made of phosphatidylcholine and sphingomyelin. Let me explain sphingomyelin, that what sphingomyelin is, and it has also a clinical rule. Okay, this phosphatidyl serine or this phosphatidyl inositol, for example, this is a blood vessel. Okay, for example, this is a blood vessel, and blood vessel is made of endothelial cells. Okay, when endothelial cells are injured, okay, in this time, this phosphatidyl serine normally is in the inside of endothelial cell membrane. When they were injured, now phosphatidyl serine appear on the outside of the cell membrane, and we have here platelets. Okay, we have here platelets, and also we have here a lot of factors which are called coagulation factors. Okay, these coagulation factors will be activated by these negative charges, so it causes blood coagulation. So that's why this is a little bit important. But anyway, let me talk about sphingomyelin. That what is a sphingomyelin? So easy. Here, instead of glycerol, okay, we have here, instead of glycerol, we have here another group of alcohol. Okay, which is the long chain, which has a long chain of carbons, and it's 18 carbon. Now, this structure, which has 18 carbon, this is called sphingosine. This is called sphingosine. Okay. Now, when we add a fatty acid, to sphingosine, so as a collectively, these two are called ceramide. These two are called ceramide. Okay, but when we add another group, it's nitrogenous base. Specifically, which nitrogenous base? Choline. Which one? Choline. So as a collectively, sphingosine plus choline, all of them are called sphingophospholipid, and we have a lot of sphingophospholipid in a neuron cell membrane. Okay, and it has role in transmission of signals, in transmission of depolarization and repolarization. So all of these were related to the phospholipid. Okay, now the next type of lipid that we have in a cell membrane, it is cholesterol. Okay, now cholesterol molecules are arranged between these feet or tail parts of phospholipids. All of them are cholesterol. Got it? Now we've studied about phospholipid, we've studied about cholesterol, we've studied about sphingolipids or sphingophospholipids. Now let me tell something about glycolipids. When we add okay, some, for example, this is a carbohydrate. When we add carbohydrates to the lipids, these are called glycolipids, and there are some functions that are a little bit important, and we'll talk about them under the title of carbohydrate. You got it? Now let me talk about proteins of the cell membrane.